and we'll move on from this particular slide. And this doesn't, you're not seeing doubles. This is the same slide that we saw before. We just want to talk about it a little bit more. We did describe what the capillary hydrostatic pressure is, but there's a few other pressures at play here. You have blood colloidal osmotic pressure, blood colloid osmotic pressure. So what that is the solutes you find in blood. That pressure is mostly made by the proteins that are floating around in blood. We call them plasma proteins. That's your albumin, your fibrinogen, and your globulins. They're too large to pass out, generally, pass out of the blood. So what they do is, because they're there and they are floating around in the blood, they increase the number of solutes in the blood at that given area. And so what that will do, because we know osmosis is the movement of water through a selectively permeable membrane, that will actually cause that water to come back into the blood volume. So the water filters out, the plasma water, so the water and plasma will filter out and then pull back in because of those proteins. And then you have the opposing pressure, I guess you might think, you might look at that's pushing against the vessels. You have an interstitial fluid colloidal pressure, osmotic pressure. So those are the number of solutes that you have in there, and those solutes can be carbon dioxide, it can be waste product, it can be things that the cell has put out, it can be bacteria, it can be pathogens, and that you have that osmotic pressure that is going to move blood, move fluid, move water out of the blood into the tissue and vice versa. So they're actually opposing each other, as well as an opposing interstitial fluid hydrostatic pressure. And again, this is a passive process, these things moving out, whether it's filtration, diffusion, or osmosis. So these pressure gradients have to be in the proper, or in the optimal ratio in order to maintain homeostasis. Going back to, that's what blood pressure does. Not only blood pressure, but the viscosity, the makeup of blood, and also the amount of the fluid, blood volume. All of these things are very, very tightly controlled by the body. There are a couple of mechanisms, and we'll talk about those as we come up, because these pressures, you actually aren't talking about very, very, very large differences. So if you, with that particular idea in mind, you have, again, if you look at the difference between pressures, when the blood is coming to the arterial side of the capillary bed, the pressures inside the blood are higher than those outside, so that will cause the movement out of the capillaries. So movement of fluids out of the capillaries. Fluids and then things suspended in the fluid, whatever can fit through the endothelial cells. And then when you get to the venal side, and this is of the capillary bed, the fluids are reabsorbed. And osmosis is most important with regard to reabsorption. So that's something that you want to put in your head. Osmosis is most important when it comes to reabsorption. That reabsorption doesn't take place unless the blood colloidal pressure is lower than that's in the interstitial fluids in terms of pressure. So these pressures have an interplay. One of the things that you want to take away from this is certainly the terms. So they're not, they don't totally They don't totally baffle you if they show up on the test or somebody talks about it. And also just the general importance of blood pressure, how important it is to maintain a normal blood pressure for many reasons. We always think of it as hypertension, hypotension, and we think of it as maybe causing headaches or causing other problems, but we don't think of it as this local capillary bed area. And remember back to that image where you have where most of the blood lives its life is in these capillary beds. I was referring to the image of the geography of how long the blood is in the aorta, how long the blood is in the superior inferior vena cava, compared to how long the blood is in the capillary beds. Then a little bit more about these processes: diffusion, filtration, hydrostatic pressure, and reabsorption. So with regard to diffusion, water ions, small molecules, will diffuse through the endothelial cells or through the fenestrated capillaries, depending on what you're diffusing through. Large compounds can only pass through fenestrated capillaries. Plasma proteins, just kind of a short list, can cross in the sinusoids. Sinusoids are similar to flattened fenestrated capillaries, so you have large plasma proteins if need be. With regard to filtration, that's where hydrostatic pressure comes into play, because filtration is the movement of or the elimination or the selection of solutes as a liquid moves through a filter, and it's based on size. Generally, the larger solutes stay in the bloodstream, the smaller ones are going to go into the interstitial fluid. And then, as I said, reabsorption is most important with regard to osmosis is most important for reabsorption. So the colloidal osmotic pressure of blood needs to actually be greater than the interstitial pressure, so those fluids can come back in. We don't want the fluid staying in the tissue. We want some of it staying in the tissue, but we don't want the fluid that we just dropped off via our blood flow staying there. We want it coming back into blood volume. We need blood volume to maintain in a normal range. The interplay between filtration and reabsorption: hydrostatic pressure forces water out, osmotic pressure forces the water back in, or pulls the water back in. So filtration or hydrostatic pushes the fluid out of the blood. Hydrostatic pushes fluids out of the blood. Osmotic brings it back into the blood from the interstitial space. Let's see, anything else that I think I need to add to this? All of these processes function with regard to, say, plasma, the plasma proteins, lipids. It certainly assists in the transport of lipids. I might talk about that a second, because remember, lipids are absorbed into the lymphatic system. So with regard to lipids, they diffuse through the endothelial plasma membranes, and they are then picked up generally by the lymphatic system. Not all of them, but because the cells need lipids too for their cell membranes. It also assists in the distribution of nutrients, carries toxins back to be taken out of the system, and also assisting with the lymphoid vessels. What you have here in this image, I guess I've kind of ignored the image here, you have a big arrow defining the capillary hydrostatic pressure, pushing that fluid out through the junction between endothelial cells. So we're not just talking about the fenestrated, we're talking about endothelial cells. Certainly larger particles would go through the fenestrated capillary beds, but most of the capillary beds in the body, remember, are these endothelial cells. Pushing that out, smaller solutes will move on out, and then as the blood moves along, because the solutes have moved out of the blood, then there are fewer solutes in the blood than in the interstitial tissue, so the liquid will move back in. Other ways that, we talked about the need for this to be regulated. Now let's talk about ways that it is regulated. So you have many major ways, or three major ways, I guess you might say. So you have autoregulation. That would be, and I apologize for this picture. I don't exactly expect you to see this picture at home, but you do have this picture in your textbook, so you might want to open your textbook. In the newer edition, I have it at 2112, short and long-term cardiovascular responses. So you might want to get that open so you can see that. And these are called concept maps. I tend to like the concept maps for study purposes, but a lot of times if you try to address the information via the concept maps without learning something about it, they're a little bit confusing. But once you have learned the information, meaning you've studied it and read about it, going back to these for getting ready for the test are excellent. Not that I'm going to ask you all of the stuff that are on these, but the concept maps tend to put the information there in one area for you. So autoregulation, you can think of that as just local. Local immediate adjustments with regard to say solutes or osmotic pressure, etc. You have neural regulation. This is nervous system. It can respond quickly at specific sites, but it can also be more long-term control. And then endocrine mechanisms. We definitely think of endocrine as long-term, which we've already defined. Neural as short-term, endocrine as long-term. But don't forget, your nervous system controls everything. So the nervous system is going to control the endocrine system. And things that will shift the regulation. So you have local vasodilators. So you have things that can just constrict or it would be expand the lumen of areas. And it looks like I'm running out of time again. I'll have to pick this up on the next one.